Very good morning, sir.
మీ వాళ్ళు ఫోన్లు టీ అంటే ఎవడు అసలు వదలట్లేదు. ప్రేమ్ టీ అంటే ఎవడు?
Hello.
Sir. Sir, చెప్పండి sir.
Okay. You are all there. So this it is comment point. That's what I called.
We all are there.
Yeah.
Sridhar.
Madam.
Please join the students when they're coming. They're waiting. Some people outside.
Yes, ma'am.
Please join. Since you are also given the co-host. Please join.
Yes, ma'am.
Join aira. Principal madam join aira.
Yes, ma'am. Madam is there, ma'am.
Okay.
Shanti.
Got it?
మేడం అన్మ్యూట్ చేయండి మేడం. విజయనిర్మల మేడం.
మ్యామ్
చెప్పండి.
రిజిస్టర్ సార్ అన్మ్యూట్ చేయమన్నారు మరి.
మీరు మాట్లాడుతున్నారు.
అన్మ్యూట్ చేశాను.
ఆ.
నేనా?
మీరు ఇప్పుడు మాట్లాడుతున్నారేమో కదా.
అది నేను బయట మాట్లాడుతున్నాను, ఇక్కడ కాదు. సరేనా? ఓకే. ఉన్నారా? అశోక్ సార్ ఉన్నారా?
ఎవరు మేడం?
అశోక్ సార్ ఉన్నారా అంటున్నా.
అశోక్ సార్
లేదు రిజిస్టర్ సార్.
ICMR वाला भी आया।
Hello, I am here.
ఎస్ సార్. అంజనాదేవి మేడం ఇంకా జాయిన్ అవ్వలేదు. చూసి
మెసేజ్ పెట్టకపోయినా?
పెట్టాను సార్ ఆల్రెడీ. ఓపెన్ చేస్తాను అన్నారు.
I think so. Maybe ఆమె లెక్చర్ ఆ టైం ఉందని అప్పుడు జాయిన్ అయితేమో.
No, she joined. I have to
join.
Yeah.
Hi, Dr. Anjana.
Anjana,
Good
madam.
morning.
Madam. Anjanadevi madam.
Sir, good morning, sir. అరే పంపిరా, రిపోర్ట్ పంపురా. వెళ్ళిపోయిందా? ఓకే. సరే వాడు చంపుతున్నాడు ఫోన్ చేసి చెన్నై నుంచి. సర్వనాన్. వాడికి పేపర్స్ లేవు కదా పాపం.
రాజేంద్ర సార్ మ్యూట్ చేయండి.
పద్మావతి మేడం, డాక్టర్ పద్మావతి.
ఎస్ మ్యామ్. మేడం అంజనాదేవి
మేడం,
మేడం జాయిన్ అయ్యారు కానీ అది అన్మ్యూట్ చేయాలి మేడంది అవ్వట్లేదు.
ఒకసారి వెబ్ మాస్టర్ శ్రీనివాస్ సార్, నేను చేస్తాను.
ఓకే.
Hello.
Madam.
Hello, good morning, madam.
Yeah, good morning.
Anjanadevi madam.
Yeah.
Okay, ma'am.
Yeah, it's audible now.
Yes,
Yes,
madam.
ma'am. Yeah.
Yeah, you had not unmuted, so I was unable to speak anything.
Okay, madam. Now it is okay, no, madam, for you?
Yeah.
Okay, thank you.
Padmavati madam, it's already 10:20.
Yes, sir.
మురళీమోహన్ గారు, వీఆర్ వెయిటింగ్ ఫర్ వీసీ సార్.
This is sir. Okay, actually ఇందాక జాయిన్ అయ్యారు మేడం.
ఇప్పుడు అది ఉంటది.
మేడం, వెబ్ మాస్టర్ ని బాలాజీ సార్ ది అన్మ్యూట్ చేయాలని చెప్పండి, అవ్వట్లేదంట.
బాలాజీ చంద్రమౌళి గారు అన్మ్యూట్ లోనే ఉన్నారండి.
మరి ఆయనేమో అదే అన్మ్యూట్ అవ్వట్లేదు అని అంటున్నారు సార్. హలో బాలాజీ సార్.
సార్ అన్మ్యూట్ లోనే ఉన్నారండి.
నాకు కాల్ చేసి అవ్వట్లేదు, అన్మ్యూట్ చేయండి అని అడుగుతున్నారు.
చంద్రమౌళి గారు సార్. హలో.
బాలాజీ సార్.
Balaji sir.
Either sir. Hello.
Okay sir. Okay. Now
it is audible, sir.
Yes sir. Yes sir.
Yes, sir.
You are
right.
very much audible, sir. You are very much audible.
Sir, thank you. Good morning, sir. Good morning, Murali Mohan garu, Sridhar garu, and Padmavati madam, HOD garu, all of you. Okay, sir. Good morning to you all.
Okay sir. Good morning.
Right,
Good morning, sir.
right, sir. Right. Good morning, sir.
Good morning.
Laya is somebody has raised the hand. Please check that. No, webmaster sir, Srinivas garandi.
మేడం
ఎవరో హ్యాండ్ రేస్ చేశారు, కొంచెం అది ప్రెజెంటేషన్ టైంలో ఇబ్బంది వస్తుందా, ఒకసారి చెప్పరా అది. The departments of life sciences, aquaculture, biotechnology, biochemistry, and zoology.
Sir, good morning, sir.
Oh, good morning, Dr. Ramneshwar garu. We'll start.
Yes, sir. Dr. Vijayanandla, madam, please start the program. Hello?
Madam, I'm starting.
Yeah.
Sir, a very good morning to one and all. Respected Honorable Vice Chancellor, Professor Mokka Jagannatharao garu, Registrar of the University, Professor T. Ashok, Principals of University Colleges, Principal of Science College, Dr. K. Ramaneshwari, and other principals, and speakers, Dr. Rajendra Rao and Dr. Anjani Dev Madam, Deans, Special Officers of PG campuses, AC members, faculty friends, and other faculty members, scholars, students, and other invitees. Hearty welcome to you all to this one-day webinar on recent trends in metabolic disorders and an epigenetic approach. The departments of life sciences includes 4 departments: aquaculture, biotechnology, biochemistry, and zoology. We, the faculty of departments of life sciences, had been organizing academic programs for students, especially in order to keep them focused towards the academics. With the support and encouragement of Honorable Vice Chancellor and other higher authorities, I'm happy to inform you that we have organized one guest lecture recently and also workshop in the month of February. Today's webinar on recent trends in metabolic disorders is organized in order to create awareness among the participants about the metabolic disorders that leads to the development of the non-communicable diseases such as obesity, diabetes, cancer, etc. Metabolic disorders prevalence is increasing at an alarming rate all over the world. Today, we have 2 eminent speakers, Dr. Rajendra Rao and Dr. Anjana Devi Madam, to enlighten us about the recent researches in metabolic disorders. I hope that all the participants will get a clear picture about the metabolic disorders and an epigenetic approach regarding how the diseases have been prevalent. I thank the co-convener, Dr. I.J. Padmavati, for her initiation, support throughout this organizing this webinar by initiating it, and other faculty members and non-teaching staff who had been there in supporting me to organize this one-day national webinar successfully. I thank all the other officers and EC members for attending this inaugural session. I thank my faculty colleagues from my own department also and other departments for the support and for attending this inaugural session. I once again thank Honorable Vice Chancellor and Registrar Sir and Principal Madam, Dr. K. Ramaneshwari, for their continuous support and extending the whatever facilities required. Not last but the least, I would like to express my thanks to the web section of the university and PRO who had been helping us to arrange these facilities. Thank you one and all. Now, now Dr. Balaji Chandramouli, sir, from Department of Biotechnology, will introduce our Honorable Vice Chancellor. Over to you, Balaji, sir.
Okay, madam. Thank you, madam. Thank you very much. Good morning to one and all. Am I audible, madam?
Yes, sir.
Okay, ma'am. This is Dr. J. Balaji Chandramouli, madam, from Department of Biotechnology. It's my privilege to introduce Professor Mokka Jagannatha Rao Garu, Honorable Vice Chancellor of Adikavi Nannaya University, Rajamahendravaram. He has done his education both in India and abroad. He got his master's degree in engineering from University of Alaska, USA. He got his PhD from Andhra University, Visakhapatnam in the year 1985. He joined as an assistant professor in the Department of Biotechnology, Andhra University in the year 1986 and later promoted to professor and head of the department from the same department. He is a serious researcher and academician having more than 33 years of experience in both teaching and research. He has published nearly 150 papers in both national and international journals. Almost 33 scholars got awarded PhDs under his esteemed guidance. His students are not only from geology, but also from computer science, geoengineering, geophysics, and allied physics. He has a strong research collaboration with various organizations from USA, Canada, and Australia. He also completed 8 funded projects from leading funding agencies of our country, including DST, UGC, CSIR, DOD, APMDC, DMRTUF, MOES, ISRO, and ONGC. He got a huge consultancy projects from ONGC, which includes studies on Holocene evolution of Mahanandi Delta, studies on land subsidence due to hydrocarbon exploration, and exploration at KG-PG Basin, Andhra Pradesh, using remote sensing and field studies. He has got vast experience in administration. He held various administrative positions like Director for Information Management Center, Andhra University, Visakhapatnam, Andhra Pradesh. Dean for PG and Professional Examinations, Andhra University, Andhra Pradesh, Visakhapatnam. Director for International Student Affairs, Andhra University. Director for Delta Studies Institute, Andhra University. Chairman for Board of Studies, Department of Geosciences, Ambedkar University, Srikakulam, Andhra Pradesh. Head of the department, Department of Geology, Andhra University, and many others. He has filed a patent entitled as an inventive model that explains the genesis of Bay of Bengal and Arabian Sea, which is under consideration. He got many awards and honored, including Dr. Sarvepalli Radhakrishnan Award, the Best Academician Award of the year 2019 by Andhra University. He also awarded Scientist of the Year 2017 by National Environmental Science Academy, New Delhi, India. He was in editorial board of Indian Journal of Environment and Eco Planning. He is the member of various professional scientific bodies of national and international repute. He is the NAC assessor and many more. He is a member of various boards of selection committees at national and state level. His earlier selections includes UPSC Civil Services, Group B Service, UPSC examinations in 1985, UPSC Geologist examination, All India 11th rank in 1983. Because of his passion for teaching, he is in the teaching life. His vision is to see that Adikavai Nannaya University should be the international institute of choice for education, research, training, and consultancy. So by this, I would like to invite our Honorable Vice Chancellor, sir, to start this program. Thank you, sir. Thank you very much.
Thank you, Dr. Balaji Chandramouli,
Sir.
for your nice words of introduction. I'm extremely happy to be part of this webinar entitled Recent Trends in Metabolic Disorders, an Epigenetic Approach. I'm extremely happy to see Dr. P. Vijayanamala, head of the Department of Life Sciences, who is conducting this webinar. Also, I'm happy to see our college principal of College of Science, Dr. Ramaneshwari, and especially we have two resource persons, Dr. K. Rajendra Rao, Dr. P. Anjana Devi. I'm very thankful to both of them for coming over here, sharing their knowledge on this subject. I'm also happy to see all my colleagues here, principals, deans, faculty members, and executive council members, Dr. Jagannath Reddy and Professor Ramesh, and Dr. Mata Reddy, and the faculty members of the Department of Life Sciences. This is yet another program being conducted by the faculty of the Kavinanda University, especially College of Science. And in this pandemic, Corona pandemic, we have been active in continuing our academic pursuits, whether it is online classes or webinars or invited talks or faculty development programs and other related activities. So I'm very happy that our faculty are very active and doing all that is necessary to continue the academic activity despite the pandemic. The pandemic crippled all of us and made us to stay home. We are unable to do the academic activity in a physical manner. But I always say that the Corona has given us an opportunity to explore the other alternatives. We never used these platforms so vigorously earlier. Now you know how we can use these platforms in a better way, how best we can see our students and the people who seek this education can be benefited by these platforms. And also we can realize and compare different platforms for their efficiency, for their usefulness, and for the applicability, not only in giving instruction, but also we need to see whether we can use these platforms to extend the laboratory exercises, because we are being scientists. The laboratory exercises, laboratory experiments are most important part of science education. So we need to explore that also. Is there any virtual experiments available? Is there any videos of laboratory exercises available? we can use them and see simultaneously, along with the theory, the laboratory experiments are also given the part as a part of the instruction. When we come to this today's topic, the recent trends in the metabolic disorders and epigenetic approach. Though I am not into this topic, but every one of us interested in this topic because we are all affected by these metabolic disorders. So when you are an educationist or an academician or a well-learned person, an educated person, we need to understand the metabolic disorders because it is affecting all of us. As it is indicated, earlier we used to suffer with communicable diseases like malaria or polio or other things. And the scientists fought for all these communicable diseases, and now we are free, almost free from majority of these communicable diseases. Now, when you talk about these metabolic disorders or diseases related to these disorders, which are classified under non-communicable diseases, which are like diabetes, heart diseases, obesity, neurological disorders, even other disorders like rheumatism, etc. Every day, every year, these diseases are growing and growing and growing because of various reasons, including the lifestyle modifications, the modifications in food habits. Now we are having a lot of changes when compared to earlier times. The lifestyle has been modified very much. Majority of us are sedentary lifestyle with little or no exercise. So in this way, these health problems are cropping up because when the lifestyle is modified, the metabolism or the mechanism of metabolism in a given body naturally will change. We'll try to adjust towards the intake of the food intake or the lifestyle of the person. So naturally, whether it affects very much the metabolic activity, resulting various side effects, including the most important, the diabetes. We can see the worldwide, almost all the countries are affected by this diabetes. As a layman, what we feel is the diabetes. If you take a lot of carbohydrates, the carbohydrates are unable to be converted into energy. The glucose is not accepted by the cells because of the insulin resistance. And naturally, it will be there in the blood. And the blood glucose level will increase, causing a lot of effects on eyes, kidneys, heart. These are multiple side effects. So one metabolic disorder is affecting all the organs in the body. Even especially in the corona, people are saying people with comorbid conditions like diabetes will be affected more by the corona. Their recovery rate is far less than the healthy person. So in this way, these need to be studied by the scientists, studied by the doctors in association with the scientists in the biotechnology and related areas. When I see, and I gone to this literature, this metabolism, the metabolic disorders can be can be controlled or can be treated. But there are two factors, it seems, which are modifiable causes and non-modifiable causes. So modifiable, maybe the scientists should think about how we can modify certain things which can control at least some extent and regulate this metabolic disorder. Mainly the causative factors, the risk factors include the age, and people growing will have more problems, and people with obesity, overweight, they have more problems of this, and other causes like non-alcoholic fatty liver, NAFL, and other things. When you talk about these things, I also, being a researcher, I wondered why certain ethnic groups are less affected by the diabetes. Because when you talk about the metabolic disorder, it should be same for everybody, but it is not like that. Some of the ethnic groups are less affected. Especially I have seen some of the countries like Ireland, Lithuania, Estonia, and other countries having less than 4% diabetic affected people when compared to the rest of the world. Is far less than the average of the world population. So the scientists need to study the lifestyle or the composition or the mechanism of metabolism of these ethnic groups. Why they are resistant? Is there anything to do with their lifestyle or with their food habits or with their genetic structure or anything like that? So in this way, it is a research opportunity for the scientists to do that way. Another thing, I always wonder, when a diabetic person goes to a doctor, he will prescribe some drugs. And if drugs will continue for some time, still the sugar levels or glucose levels will increase 200 or 300, then immediately you venture into go for insulin. So as a scientist, I wonder why a patient is not studied in depth about his metabolic activity and mechanism. Because each and every person will have a different types of metabolic activity towards different types of food. The doctors, why they don't study in each and every person? By putting him or her in the hospital over a period of time, giving different types of foods and see the sugar levels increase and how the body is responding to different foods by a person. Because each and every individual cannot have same type of response to the food, but they are not doing it. Not that they don't have knowledge, they have the knowledge, but they don't have time. They are more interested to earn the money. They are worried about the money because they want to earn the money. That's it. So that is what I understand. Each and every person's metabolic activity differed from other person. So we need to study over a period of maybe 15 days, 1 month, 2 months, by putting him under different types of foods, then prescribe, okay, this is the best possible food for you as per your body's response or insulin resistant. Because everybody's pancreas may not work in a similar way. I have my pancreas working at 30% level or 40% level. Somebody is 60%, somebody is only 10% is affected. So why you give say metformin XYZ and after some time you give insulin? In that way, you are spoiling the health of the person. That is my personal opinion. So in this way, All these things we need to study because this is the biggest problem. Maybe this problem will continue in future, and you, the scientists, have greater role to work and come up with the best possible solution. So that is the greatest help to the mankind, so that they'll be happy and they lead happy lives. So you have the responsibility to do research. As you have indicated, we need to work on these epigenetic pathways. Thank you very much. Thanks for giving me this opportunity.
Thank you very much, sir, for your message to the participants. Thank you, sir, for briefing us about the metabolic disorders, and also giving us the detailed picture of how the insulin is being produced, and what are the steps, what are the research to be taken by the scientists. You reminded us, sir. Thank you very much, sir. And once again, we thank you, sir, for encouraging us to work even during this pandemic situation to focus towards the academic side. Thank you, sir. And I think all the researchers, those are in the group, will take part the studies on certain groups who are resistant to diseases, so ethnic groups. So I thank you once again, sir, for your valuable message and sparing your valuable time and joining us today for this inaugural session. Thank you, sir. Thank you, one and all. Over to today's co-convener, Dr. Ijen Padmavati.
Good morning to one and all. Honorable Vice Chancellor, Professor M. Jagannath Rao, sir, respected Registrar, Professor Ashok, sir, respected Principal, University College of Science and Technology, Adikavi Nanna University, Dr. K. Ramaneshwari, Madam, respected other principals, deans, and officers of Adikavi Nanna University, respected invited speakers, Dr. Rajendra Rao, sir, and Dr. Anjana Devi, Madam, respected faculty members of life sciences and other departments, my dear colleague friends, and my dear students and other participants. Welcome you all for this national webinar on recent trends in metabolic disorders and epigenetic approach. It's my great pleasure to introduce today's invited speaker, Dr. Rajendra Rao, sir, scientist from ICMR National Institute of Nutrition, Hyderabad. Dr. Rajendra sir obtained his master's degree in genetics from Osmania University and a doctorate also from genetics department, Osmania University. He has a vast research experience since 1999, worked in many national or research institutes such as Directorate of Rice Research, DRR ICAR, Institut National de la Recherche Agronomique, France, University of Miami, USA, CDFD, and also the group had molecular biology and immunology in BPRC, Lepra, India. And he joined in National Institute of Nutrition as a scientist in 2006. He received many honors and awards and invited participants for Ideal Workshop at Bangkok, Thailand. He awarded Sastry Memorial Young Scientist Award, ICMR International Fellowships for Young Biomedical Scientists in 2007. He is a visiting scientist for the Cancer Institute of New Jersey, USA, and also visiting scientist, the Rockefeller University of New York, that is Howard Hughes Medical Institute, New York, USA. He is a member of several scientific societies like International Society for Genomic and Evolutionary Microbiology, who is a life member, Nutrition Society of India, again life member, and Indian Pharmacological Society, the Epigenome Envoy, Laboratory Animal Scientists Association, and BYS member of Department of Biotechnology, Osmania University, and St. Anne's College for in Hyderabad. He got many research publications, published more than 40 research articles in peer-reviewed journals, contributed to 6 book chapters, and as a co-author for 1 book, also as a reviewer for 10 journals. He guided many students and scholars. Presently, 6 students are pursuing their PhD under his supervision. He executed many national and international projects funded by DBT, ICMR, NIH, UKRA. He executed many national and international projects. His research interests mainly lie on impact of nutrition on non-communicable diseases, that is genetic and epigenetic approach. With this now, I invite Dr. K. Rajendra sir to deliver his talk on diet, gene and obesity to today's national webinar participants. Thank you. One and all, over to Dr. Rajendra sir.
So, good morning. Thank you very much,
sir.
Dr. Padmavati, for your kind introduction. First, I would like to thank the VC sir. He made my job easy, giving a wonderful inaugural talk. Sir, thank you, sir. Yeah, non-communicable diseases. You told that what are the different types are there. Some can be reversible, some cannot be. And also, you touched a very important point, sir, that ethnicity is really very interesting. Where also, every time I'll emphasize on the things, because no, this is by keeping in this only. I started working on the nutrition. My research here. When I come to the NN, I don't know what I have to do in NN, but because no genetically modified foods and all was actually what I applied. But after coming here, that total scenario changed. So thank you very much, sir. Very few people will think about the ethnicity and all. Whatever they give the direction, they never look into the ethnicity. So genetic backgrounds of the people are very much important. And also, VC sir said metabolic disorders and diet variation. Yes, your body response naturally it is different from person to person. People cannot understand this. So where my research is going on that line, sir. So I request you to, if you continue and listen to my, spend some time to listen to my talk. Of course, I made some general things, and as well as my research work, some specific. So now, with these few words, I thank all the organizers for choosing me as a today's speaker, and also it's an honor for me to give a talk in a national seminar for the Adhika Vinaya University. Thank you. And also, of course, happened to be Padma is my student. So all the best, Padma, for a wonderful job you are doing there. So with these few words, I would like to go for my talk. I'm sharing my screen. Is it okay? Disabled share, host disabled attendance. Please able me to share, no?
Webmaster,
Host is not
Srinivasa
allowing me
garu.
to share my screen.
Yes, sir, it is permitted. It is permitted, sir. Permission granted, sir.
Okay. Has to share, no? Some problem. Just I'm checking, please. Some. Yeah. I hope you got it.
Yes, sir. Yes, sir.
Yes, sir.
Yes, sir.
Obesity.
Yes, sir. You can see it.
Diet.
So it's visible, sir. The screen is visible.
Visible.
Please go ahead.
So, good morning. Once again, I say a couple of times good morning because I'm a nutrition man with the genetics background. This is our institution. Is one of the oldest institution in India, NA, National Institute of Nutrition. So, good morning. So I can say that no, you are all seeing the flowers. but I can say it is a colorful flower, a cheerful good morning for all of you. Everyone want to be healthy like this, like flower. So this is the idea to go for this. And also again, once again, good morning. The fruits. So I give the message for everyone, and also I request you, please every day consume a fruit.
Sir, excuse me, sir. The slides are not changing, sir. Please change the slides.
Yeah, I changed here. No. Now?
No, sir. The first slide is there, sir.
No. Yeah.
Oh, yes,
When
sir.
I am
Yes, sir.
so presentation mode, it is not moving. So anyway, I'll do this. Is it okay? Are you
Yes,
able
sir.
to see now? Yeah, yeah. That's a colorful. Whatever the flowers are there, then you can see everyone want to be healthy and happy. So that is the idea to tell about this slide. So then this is fruits. So when I say people, when I ask me, what are you seeing me? They say fruit. No, we are looking at the healthy. Your health is here. Most of the minerals and vitamins is available through vegetables and fruits. Please consume seasonal fruits and vegetables. So different type of fruits, different type of colors, and all these will give us the health. So now VC sir said that yes, during Corona time, people are looking for the immunity booster. Immunity is in front of you, health is in front of you, which is the cheap and best till today in this world. That is the fruits and vegetables. Consume it. When compared to the processed one, always we prefer eat the whatever the fresh, whatever the nature is giving. So please, this is about the healthy morning, I can say. Then come to my talk. Here you can see that no one right side, because no, it is not animation mode I put, but the thing you can see this. No,
Yes, sir.
animation is not working. Here the thing I have to go like that, right? Anyway, this is about the genetic variation, how the people will look. Everyone is having their own genetic background all. So you can see the insects all are having a different spots on this one. This we can put a question here. So
Sir,
second, here you can see the yeah.
Rajendra sir, please change the presentation to complete screen mode. Presentation mode you can change.
Presentation mode, madam. That's what no, it is not main moving.
Okay,
Now
participants
in my
are asking.
presentation. Otherwise, can I send it to you, Padma, by mail?
Sir,
Doctor,
Sir,
can you
sir,
share
you
there?
go ahead, sir.
you go ahead, sir, like this.
Participants are asking for it.
That's true, but I cannot show the animation, whatever I see. No, my lecture also will in other way, otherwise. So,
Sir,
okay,
you go
now
ahead, sir.
shall I continue?
Sir,
Yes,
continue,
sir.
sir, please, sir.
Continue, sir.
Yeah. Here the genetics, where you can see the cursor. No, cursor you are able to see. No, yeah. These are the insects. Insects are having the dots here, and also the maize is having some different colors. We can put a question here: is it the genetics or environment? A question comes. When you come to the flowers are because of the genetics or environment. So when you come for these, we can say genetic is definitely the genetics. When you see the Drosophila, these are all genetics. So when you come for this, many doubts will come. So how the genetic background with the environment is interacting and how it is going. So just to define it, then we'll see what is genetics and what is epigenetics. When environment is playing a role to change any character, a phenotype is called epigenetic. Here the genetics is probably that you have inherited from your parents. So whatever it is coming from our parents, so the genes we can say, chromosomes we can say, this is a genetics. Then epigenetics is a new area to study. This is some of the things are inherited from the parents, and also the environment is having a bigger role in this one to change the tags, tails of your DNA or your protein. So there we can say it is the DNA, but it is not changing the sequence. But DNA sequence is changing in the genetics. But whereas in epigenetics, it is not changing your DNA sequence, whereas it is changing the tails of the DNA. We can see in detail in coming slides. So that no genetics, you can say it is giving the phenotype. You can see the yellow mark here is the star. It is a mutation. a change, then we can see the polymorphism, then we can see a SNP, that what we can say. In this case, we can see epigenetics on the DNA double helix, you can say a spot. It is epigenetic mark. It may be permanent or may not be permanent. So this is about the genetics and epigenetic differences. Right. So then what is genetics? Again, I want to put the question. What is genetics? Here the question is that all of us studied the Mandalas in our childhood, 7th, 8th class, like that. So it is a branch of biology where compared to the study of the genes, genetic variation, heredity, inheritance, all this will be involved in the genetics. So that the best example, whatever we studied is the Mandalas. So Mendel is classed with the Poisson satin with the white with the purple or round seed with the flatted one. So like that, these are all the things we studied where we got all the basics. So that man, we are all having know that yes, something is coming from the parents to the children, offspring. So the cell is carrying. What is that material is carrying? So earlier people know that some material is coming from the parents to offspring. Then we come to know that there are chromosomes. Then the research said that no, yes, inside the chromosome something is that is a genetic material, that is a DNA, deoxyribonucleic acid. So that when the DNA is in the chromosomes are in the cell. The cell is building blocks of your body. So then it is very simple to say what is the composition, what is the language of the genetics, the genes. It is having only 4 bases are here: thymine, cytosine, so adenine and guanine. These 4 letters are playing a role. Like English vocabulary, we can see only 26 letters are making many words. But here also same way we can see your 4 letters are deciding your health or the disease. So how you look, the phenotype. So this is about the brief background of the genetics. So functional genetics. What is the meaning of functional genetics? Whatever the total DNA which is present in your body, it may not be functional. So the part of the DNA or the genes are working. So that is called a functional genomics. In that, functional genomics is understand the relationship between an organism's genome and its phenotype. Phenotype means how you look is called phenotype. So my phenotype is a healthy phenotype. My phenotype is a cancer phenotype. My phenotype is a obesity phenotype. I am a diabetic phenotype. That is called phenotype. How you look. So that it is a functions. What is functioning inside? The functioning inside is a genes. So genes, when it is speaking something is telling, so that is it is giving the RNA. RNA is giving a protein. So that is a language of the molecular biology. Central dogma is a DNA to RNA, RNA to the protein. This protein is deciding how you look. So here the functional genomics means yes, your gene is okay, whatever the nature is giving, it is giving is accurate, then it can form. If any change comes, that is a genetic mutation. So whatever we are speaking in this protein or RNA, DNA, these are all called a molecular biology. So we are speaking about the molecules. So this is a molecular biology where you can tell lot of data is there. People are going. Earlier they studied at chromosome level, how the chromosomes are working and all. So then we come to know the chromosome inside DNA is there. Now we know the DNA gene language. So then now the technology, we can cut the DNA into the pieces. That is a marker. Now we can read the what is there in the DNA. That is called a sequencing. So the sequencing technologies now we are using at present, we are using high throughput techniques. When at a time, several thousands of the genes we can do. Now the corona, whatever the SARS-CoV virus is there, your VC sir said, yes, every day we are sequencing. Throughout the world, people are sequencing and they are comparing and they are telling the Delta variant, Beta variant, whatever the different variant. For your information to tell about this coronavirus, it is a retrovirus. The retroviruses will undergo for a lot of change. So that it is a natural process of its survival of the virus. It has to survive in the nature. So that no, these are all happening within no time. Now we are sequencing all this because the technology is increased. So on that DNA basis, we can say when we are screening many genes, it is called microarrays. So when we are going for the many proteins, it is a proteomics, studying about the proteins and epigenome. Just now I said, not only the DNA sequence is not changing, protein sequence is not changing, but the tail of that one, acetylation, methylation, it is somylation, these are all changing. So when these are all changing, then it comes under the epigenomics. Then a protein part, Dr. Anjani can speak, and the DNA part I am going to speak. Then metagenomics. So already existing literature is there in that we are all digging and all the analyzing and doing all this metagenomics, or unknown genomes we are taking. Under the sea, many type of organisms are there we are bringing out, or you can take from the gut. Several type of bacteria will come out when you are doing the endoscopy and all. When you are sequencing all those things, we don't know what is a species and all. These are all comes under the metagenomics. Metabolomics, how the protein is deciding your metabolism. Today we are speaking of metabolic diseases. Mean because of all these actions, your metabolism is changing. So when you are studying about all this is called metabolomics. Where metabolism is changing because of the mutation. Where the mutation is coming at DNA level, a change is coming. When the DNA level change is coming, the protein is changing. So this way we are going to know all the functions. That's what it is called as a functional genomics. Then where the change is coming. In the DNA sequence, you can see the best example is here. Again, A, G, T is there. So the sequence of this, the changing of the one letter. So it is changing your protein, your amino acid. So you can see to schematically to explain, this is the color of the person we can see here. Yes, changes come. Because of in this one, it is changing the protein, your function is changing, your metabolism is changing. So that it is called a single nucleotide changes called a single nucleotide polymorphism. In this, total DNA is having the same functions? No. Wherever the DNA is having the function, it is called as a gene. So that is called, in that gene, exon part, it is undergoing for a change, it will change. Or promoter is undergoing for a change, it is. Or intron can be also said. Sometimes it is having some effect. But non-coding part is the intron, whereas it is also having the effect on the your function of the genome. So these are all showing the effect on the your genome. The genome is affecting in the phenotype. Phenotype means how you look. So this means these are all linked to the all your body metabolism. So then we can see here the coding, non-coding and all, it comes the protein. So individuals you can see in the profile of the SNPs, we are all looking the same, for example, where I showed the pink color. So in the course of the evolution, so some people are becoming diabetic, some people are becoming hypertensive, some people are becoming cancer, and these are all changing. How these are all changing? Because your environment or because the living habits are there. This is one part. And natural mutation sometimes, when you are exposed to the high radiation and all, so that is also taking a place. But whereas we are the multicellular organisms, in the case of the human, and so then that is not that much easy to change. But even though during the pregnancy time, a gonads can undergo for change, a sperm can undergo for change. So that concerning all those things, individuals are having different phenotypes. So we have to study about all these things. A change can be at the DNA level. So that is having a change in the phenotype. So is a genomic revolution has come. When the genome in 2000, they have taken up a big project, world big project is a genome sequencing. So that they sequenced the genome, they published it in 2001. Human genome totality sequenced. Then that time they thought we can challenge the God. We challenge the nature. But still we are looking for the things, many things we are learning. So how many are functional, how many are not functional? Why this much of the genome content we are having? These are all we have to see. So now the high throughput genotypes technologies are giving at a time we can screen many genes, and that here biostatics are very much important. You have to validate whatever you are doing. It is statistically, it is acceptable or not. For that to do, first step is whatever the data comes, we do the bioinformatics by using the computers and all. We are doing the big data analysis nowadays. We know that a big data analysis is going on, so that then we validate all those things by using the different formulas and all. So then it comes when we assign some function to a part of the DNA. It is any functional gene, a candidate gene. So that is about the functional genomics. So then how to identify? Of course, telling all these things is easy. How to identify a gene? Your genetics is different, my genetics is different. I can put a simple question to you. So within the family, my family is in this, we are the 5 brothers. My mother and father is, so there, then we are the 5 brothers, but we are not looking the same, except is identical twins. Why we are not looking the same? Parents are same. Some change is coming because of that, no? So when I get the cold, my brother will not get. When I get the fever, my brother will not get. One brother is a diabetic, one brother is a hypertensive. So these are all happening now. Of course, we are all now in more than 50 years. All are. So that no, why? So these are the things we need to consider. So that here the genetic background of the person is very much important. How to identify all this? Changes are coming, SNPs we said, high throughput technologies is there. That's what now we do all the linkage studies. A linking mean according to the character in the total population. It is called as a panmictic population, a randomized population. We pick randomly some samples we take. In that characters, how many are there having? Accordingly, then we can go for the genome linkage. First, we identify where it is linked and which chromosome is linked. After that, no, then we can map it where exactly and which chromosome it is falling. After mapping that part of that one, we can go for the sequencing or genotyping. After sequencing that, then we are going to know where are the changes come. So by statistical analysis and all, by using the bioinformatics, then we can see is conventionally they did only link. Then the part where it is there. Now we are tuning this one and we are identifying where exactly which location is there. My work also I can show. So on that basis, we are identifying the candidate gene. Yes, this gene is linked to the diabetes. This gene is linked to the hypertension. This gene is linked to the cancer. Like that now we are telling all the things where the change is coming and which gene is undergoing for a change. So that genetics of the person to person will vary. So just to classify a syndrome, many diseases are together, it is called as a syndrome. So for syndromic things, there is no medicine for it. Of course, it is symptomatic they give. And monogenic disease, when only one gene is undergoing for a change, then color blindness is there to identify this one. So that no, these are all monogenic. One gene can change and it is giving. How this theory has happened in 1902, the Gerard, the scientist, the physician, is identified phenylketonuria, PKU disease. When people will go for the toilet, when they are pissing, so the toilet will turn into the black. So some changes come. He said some error is there in the at base level. They don't know about all the genes and all the things. Now we know that where the change is coming. So in phenylketonuria is because of the gene is undergoing for a single mutation that is giving a change. So that where instead of the gluten, the valine, we can see this. So in this one amino acid change is giving the total change. So that leads to the disease. Like that, now many monogenic diseases are there. Then polygenic diseases. What is polygeny? Many genes are associated with that. To tell one example, is that diabetes is there. Diabetes is not a single thing. Of course, the pancreas is not working. It is not producing sufficient insulin is one. That is a defect of the one. The type 1 diabetes is, we can define it. Yes, genetically it is there. When you go for the type 2 diabetic, people are assigning many genes on that one. You can read my review article on that. So that now here we can see several genes are playing a role on this one. But diet is there, environment is there, living style is there. Many things are there for this. So that several genes are when associated with one condition, then it is called a polygenic disease. So this is the types of the diseases and all. How we are going to come for a conclusion? In a family, it is happening. How to go for that? Whom they married to each other. So I will tell the best example is the color blindness. The color blindness is carried forward to the next generation. In that, who is having the color blindness? Mother or father? So that no, whether they are a carrier or they are showing the symptom at that character at itself. So on that basis, we come for a conclusion. So that no, it is a monogenic disease is going from the generation to the generation. This is called genetic counseling we do and genetic calculations we do, and we tell this is called the counseling is very much important. Any family is having the muscular dystrophy or color blindness or this type of single monogenic disorders are there. Please, I request them to go for the undergo for this type of counseling, and they are going to know that what is the next generation is going to affect. What are the chances for that one? So where we can avoid this type of things and we can avoid the punishment for our next generation. So this is where we can use all the genetic studies to take only speaking about only the genetics, the genes and their changes. That's what I said. We 5 brothers are not same genetically. My parents are same. Because where it is changing, we studied the mitosis, we studied the meiosis. In the meiosis, it is undergoing for in the prophase. You can see that crossing over is taking place. The crossing over places are changing as the person to person. When I took birth, it is different. When my brother took birth, it is different. A little change can give a big variation. So that these are all very much important to understand the genetic variation in the people. Genetic backgrounds are not same for this, except identical twins, not the paternal twins. So then you can see the fraternal things also. So these are all I want to show. MSc students are there in this one. Nowadays we are using different technologies to go for all this to understand how many genes are playing a role, different clusters, all this forming. So this can give the new technology, new ideas to form the clusters, and these are called heat maps. So we can produce the heat maps, and in that we identify some part of that one. We can go for the gene identification. This is example for the type 2 diabetes. You can see how many genes are associated with this. So then this is to tell about this one. Just to give in a nutshell how the concept will come. We hypothesize that one. Yes, one gene is there or many genes are there. So for that, what we do, then we can go for the fine mapping of that one. I said the linkage and all we do. Then we'll map it. Then we'll identify the location on the chromosome. Then what is the gene we suspect? From that we can variant identification. That part we can see how much variation is there then. Then once we identify the gene, we can go for the functional prediction of that one by using the bioinformatics. Then we work on the in vitro studies, either rat model or any animal model we take, and we assign the function to that one. So this way we can go for the any gene of identification. Come to our work. So my work is this one. What I did when I joined in 2016 NIN. So there was a rat where the suppliers of the animals for the national research, about 470 institutes we supply the animals. So rat, mice, rabbits, so gerbils, guinea pigs, and many are there. So in that they identified one rat which is obese. So why suddenly it has become obese? They isolated, separated this one, and they started breeding this. So it is giving obese animals. What could be the reason? Because of the diet it is eating more, environment effect or diet effect or genetic effect. How to rule out? So that now they did some physiology work and all. After I joining that one, I started the genetic dissection. So what is the meaning of genetic dissection? What we do, we take the obese rat, we can cross with a normal rat, which is a lean one, and we can cross with the relatives of this one, which rat it is generated, and again we can cross with the unrelated one, a distant one, not the related one. So like that, I made all the crosses. That is what I can say here. F344 rat is a genetically distant to this one, and another other rat is called a Vista rat, the relative, the main ancestor of this one. That way we crossed and we developed the mapping population. In that population, how many are having this symptom? How many are having this character? Like that, we can link to this one. On that basis, I did all the work by doing all this. And this polymorphism is linked to the chromosome number 5. So then I isolated the DNA. I used about 160 primers, markers. So then SSR markers I used and I genotyped all those things. Then after come for a conclusion, then we can see yes, on chromosome number 5, some changes come. Where on the chromosome number 5? Then we link it to this one. So the linking part is this part, wherever we can see the red one, this here, it is linked to that part. So what is there in this one? Leptin receptor is there. Okay, leptin gene is there. In between it is falling. So still the work is going on. Once we sequence this part, then we are going to know what is what, where it is falling. It is a coding or non-coding or promoter part. So part of the work we published in the some journal is the plus one. So this way we can go for the work. When we want to identify something which is new to us, this is an example to say my work is this one. So then here, thanks to the ICMR for giving the lot of funds and also NIH America for this one. So this is now the new technology come to tell again to the MSc students that CRISPR-Cas9 technology. You want to create any mutation somewhere in the gene, now the technology they are using. So you can create a gene also, knock out mice, then we can say transgenics, they are creating all those things by using this one. So then this is a new technology, whatever we are using. Okay, just a moment, please. Yeah, sorry. Yeah. Thank you. So then here it is about the CRISPR technology. Then towards the integration of the knowledge. Of course, lot many things are there. So I spoke about the Mendel rules. What is Mendel? Like classical genetics. Then whatever used for my rat is the only classical genetics I used at beginning. So that is always useful to the fundamentals are very much important. Then chromosomes, karyotyping, ideograms, and all the second stages come. After that, DNA markers we used. So that is another technology. Now we are using the large scale, high throughput technology. So in that high throughput technology, no need to do all these things, but take the DNA. What are the functional parts you put on a slide, a small slide. You can see on slide, we can put the small cover slip. So that is 1 cm into 1 cm, 1 centimeter, 1 centimeter. On that, we can put about 35,000 genes. So you can see all these. Here, all we can see, and it can read by the laser. Then it can give all the spots. On the basis of each spot, which is linked to the bioinformatic analysis, now we are doing all these things. Mean no need to wait for a lot of time. Then we can do this technology now. How we are integrating all this technology. So the technology integration is a bigger part. It is playing a role. So that now every model, whatever is there in this, the known parts, things we can take, then we can go for the first step, then we can look into the unknown things. So these are all called genomics, proteomics, and whatever the metabolomics, whatever you're telling, the computational biology helping a lot for us. So then here we can see new targets. You can see the heap of all the genes or genomic data is there. Our duty is to look into this one every day and find what is happening in this. Of course, bioinformatics people are working. So tell one example for the obesity, we can say FTO gene is there, MCR genes are there. Okay, on the basis of the BMI, MDS, these are all the some of the genes there where we can identify. Then people are identifying throughout the world and using this new technology. So come to this one. Here, a variation is coming. Just now, whatever we saw, a change is coming. How? It is there, then a DNA change, a chromosome, which is there inside the chromosome, the DNA is there. Yes, where it is present in the nucleus, it is in cell. So ultimately, any change comes, your phenotype is changing. Again, question comes for us. When we go for this one, it is the genetics or environment. So again, we have to put a question. Yes, when you come to the butterflies, you can see butterflies are undergoing for different colors are here. So industrialization of this one, where butterflies change their colors, because now it has to protect from their predators. Then in the case of this one, here the genetics, 100% genetics, because we are growing old and all, these are the variation are there. So both we have to consider to define how you look. Your phenotype is decided by the both. One is the genetics and environment. Just now we saw some examples of that one, genetic disease. One is we can see color blindness, I told. Second is obesity of our rat, I told. So how to identify the genes? These are all pure genetics. So how the environment is affected? That is called epigenetics. These are epigenetical effects. We cannot rule out that one. Genetics is not playing a role in this mean. Yes, genetics is there, environment is there, but togetherness is there in that. That's what VC sir said is yes, your genotype, what you have to eat. You have to eat the more protein food, you have to eat the more carbohydrate food or more lipid food. What do you have to eat? According to your genetics, can we go for the personal nutrition, personal diet? Now, personal medicine people are telling, is it possible? We don't know. We are in that line to find out. But whereas then things are going on. Here, when you come to this one again to show that one, the same thing I have taken here. What is genetics and what is epigenetics? Again here, your own environmental exposure and lifestyle is also affected the tags, the tails of the DNA, the tails of the protein. So here we know now genetics. Now we know about the environment. Environment is what you drink, where you live. So how much stress you are undergoing for. So then when you are in a mother womb, what your mother is eating. So what you are eating at present, these are all matter for us. So these are all deciding your epigenetics part. So that then why variation? Why variation has to come? What could be the reason for the variation? Why changes to come? A question come for us. Yes, now things are like this. We cannot change anything, but things are changing. It is changing our health also. Health and disease always it will change. It is not stable. So then, VC sir said again, sir, I will take your example again and again here. Yes, reversible, not reversible. Then you can see the food habits. All greeneries are there and slowly disappearing into the bread and pizza, burger and all the things. Now, earlier we lived like this, a greenery. Now industrialization is there. We cannot avoid it. Because no, the evolution or development, all these things, for our survival, we are doing all these things. What it is happening? It is having effect on you. So that is affecting your protein, that is affecting your DNA. So that here the thing is that the DNA we cannot change immediately for multicellular organisms until and unless any defect comes. Example, I said the PKU, phenylketonuria, then type 1 diabetes, whereas type 2 diabetes is not like Type 1 is very few, minute. So the prevalence is very less. So type 2 is a more world over. Then obesity is there. So cancer is there. These are all mean gene and environment are working together to decide your phenotype. So now we can say, yes, your genetics is deciding one factor. Other factor is your environment also playing a bigger role. That's what now. When you want to read your clock, you have to read your clock in a such a way. Yes, there is a clock. Generally, we can see in a round shape. When you see that, no, it is keep on changing. The molecular clock is undergoing for a change. Yes, is it true? Yes. Your genotype and your environment. When you come for this evolution we studied up to here, this is correct. And phenotypic is there. Why this obesity has come into the picture? Now we are defining your phenotype is obese. So when compared to this one, he is more obese and overweight. What could be the reason for it? Is it culprit is your genetics or your genes, or where you are living, or together? This question comes. So that now when you want to see an environment effect, it is called the epigenetical aspects. So these epigenetic aspects also playing a role. The togetherness is always playing a role. So for to decide your this, what is the prevalence world over? I have taken this from the WHO data in 2000. How the obesity is there in the world over, and all now new records are there. We have to insert this one, so that then we can see still India is in orange. So when compared to some part of this one green, this type of what sir said ethnicity. Why Kashmir people are not much obese when compared to this high obesity we can see in the Gujarat area and all. So what could be the reason for them? Maybe their food habits, their genetics. A question comes here. So that to rule out all these things, wherever the food habits are changing, the things are changing. The togetherness is always there. So then, here a body mass index, how to measure for this one? Your BMI we have to measure. On the basis of the BMI, you are overweight or obese. It is decided. Anyone can read on that. Now you can go to the WHO site or Google it, you can find. So obesity, if the more obesity is there, what is happening? It may lead to the development of the diabetes, type 2 diabetes. It may lead to the development of cancers. It link to the many things. Hello.
Sir.
Can I continue or someone want to speak? Yeah. Here, the diabetes prevalence has been rising more rapidly, and low and middle income countries. Mean world over, it is a big scenario that one to show that they said how they are deciding on all the things. Come to this one. What is making you wrong to become obese? What is the problem? So that is, you can see at right side, the animation is there. It was good, but now not there. Here you can see a right side, the squirrel and the garden lizard, a lion. These are all, if you ask your great grandfathers also say they are looking the same. They are not changing. Even in front of our domestic animals, the goat, dog and all, they are not changing. Why they change? You come to the left. You can see all these things. When domesticated, when you are giving the different type of food and living conditions, even your domestic dog also become obese. Goats are become obese. Your sheep also become obese. So that a changing environment is having lot of changes. Why they are not changing? This lion and all. What could be the reason for it? Is it your willpower or instinct? So we can put a question on this. So that, and also see some more examples here. You can see here the paintings of the Ravi Verma. Then we can see no one is obese here. You can say Jaruvas in Andaman Nicobar is a tribe. They are not obese. Now, of course, tourism travelers are going and they are giving different type of foods for them. So always they are saying, because our ICMR institute is dealing with them. Then here we can see some of the figures on the temples. No change. Come to this one. This is a Mysore Maharaja. So then here you can see no one is obese. Come to the Nizam King in Hyderabad. They're all not normal because they are not obese. Why? Why we are becoming obese? What could be the reason for it? So then we can see the life is a lifestyle and environment. Your diet and your lifestyle, both are working here. So then you used to walk a lot, used to work a lot. Then you started using the horse cart or bullock cart to go somewhere. And ship has come into the picture. So then motorcycles have come. So different type of this thing, and cycle has come. So then four-wheelers have come, now aeroplane has come. Physical exercise, physical activity is coming down day to day. Then also your food habits also changed a lot, drastically it changed. So that here we can see here, then we use it to consume all the down ones. Here we can see all these things. What nature is giving which to consume a lot. Now we are consuming all these things. Sugar, salt and all, maida and the Coca-Cola, whatever is there, no, soft drinks. So this change is having effect on you. So it is interacting with your genome. It is influencing your genome. Both are there. It may influence definitely. So that no habits of this one, we used to work a lot. Now machinery is working. So now flowing and all disappearing day to day. We used to cycle a lot. Cycling is a lot. Now we can see spending more time in front of this one. A good example, I took this one from the cartoons. In 1919, so that people are saying you are thin and your TV is now. So now we can see the change in 2017. Same thing, we are spending more time with the mobiles and all, so that your physical exercise is going down, what it tells. So that, are you sitting too much? A question comes. Sitting too much is really a problem? No. So the thing is, your physical activity is a problem. So that whereas our living habit also changed. Now machinery has come into the hands. Now we are working on the systems. So that now this is lifestyle is changing. What I want to say is a lifestyle is changed a lot. So how these are all impacting on a diet and lifestyle. Your food is changed, your diet is changed, whatever is available also we are not taking. We are taking the ready-made and packed foods and all, and fine-tuned foods, polished foods we are taking. So that these are all environment mean your diet, your physical activity. So whatever, then the stress also very much important. The economic stress is there, your job stress is there, many things are there. So then here we can see smoking habits, drinking habit, different type of habits. These are all having environment. These are all called as environment. They are allowing the epigenetic mechanism. So if any genetic change is there, directly we can show the effect on that. By people, some are by birth they are obese. No problem, they are healthy obese. But when your environment is changing, you are becoming obese. Then it is called obesity. It is irreversible. When you change all the things, it can come to the normalcy. But is it possible? That's what I asked the question. Is it your will or instinct? Then how it is affecting? Not only that one, whether you are when you are in the mother womb, so that time whatever mother eats also very much important. The memories carry forwarded. So Padma work is on that one. Dr. Padma is worked on that one. How the chromium is effect on the deficiency of the chromiums and mother on the children. So that this way we can see all these environmental effect is having impact on them. So that the complexities of this one, exercise, nutrition, virus, hormones, virus. Nowadays you can see in the newspaper, we are reading that one. Yes, viral infections are leads to the one. Please 1 second. నేను లెక్చర్ ఇస్తున్నాను మళ్ళీ మా లెక్చర్ ఉన్నది. So then technological progress, pollution, psychology and pressures, all these things are there, not only one. We cannot blame only one thing. Lifestyle mean many things are there. People are simply blaming that I'm eating more, that's what I'm becoming obese. No, wrong. Please keep in mind, all these things are there, multifactorial. So that no, if you can avoid all these things, I mean, how to best balance all these things, I can say, then you will be normal and healthy. You cannot become obese or diabetic. So then what is happening inside the body? What are the things are changing in you? So that's what now what we can see the signs in this one. So there systematic inflammation will increase. TNF alpha, IL-1 beta, INF alpha, gamma, IL-6, IL-18, all these things are so enhanced. Okay, so free fatty acids and LDL. So these are all increasing here. Then TG levels are going up. These are all impaired. So impairing of all these things are finally leads to the change in your fat levels. So when it is changing, the inflammation is more, the your fat size, the normal fat is like this adipose tissue, and the adipose tissue hypertrophy, you can see the bulged one. So that where we can see macrophage infiltration, these are all the things are happening. So that leads to the development of the obesity or any metabolic disease. That may be a cancer, that may be a diabetes, whatever is there. But when all these factors are having a role on this one, so then these are all leads to this type of changes. That's what now when coronavirus is infected, so doctors have started writing for the CRP levels, IL-6 levels, and also the D-dimer for the blood clots and all. Yes, definitely where the inflammation is there, stress is there. So that is having impact on your health. So that now they are writing many. So now we can see the some of the papers on that. Yes, coronavirus infection in the post-coronavirus leads to the development of obesity or diabetes. So then, so how to avoid, how to go for this also, we can see that again the same thing, it can come, go for the exercise. So balanced diet mean whatever your mind is, eat to that one. So any infections are that, rule out all those things. Then stress, you can avoid the stress. Then technology progress, we cannot do anything but taking too much of the pressure on this one also a problem. So then come to this. Here, just to say in a nutshell, this is the self and Mediterranean diet is this one. Whatever the diets are there, greenery and all, that leads to this one. And whatever the diet, processed diets are, that leads to this one. How your liver we can affect on this. Just to say one example for this one, to show that schematically I made this one to understand. What is happening, it is not the genetics. Genetically both are same, but your epigenetics, this is. Environment is having effect on this one. It is changing at your DNA level or your protein level. It is called epigenetical aspects. Then, and come to the same, again the same slide. What is happening then? Okay, genetics is defined. Yes, any change will come, it will change. Forever it is. Of course, then some medicine is there, some protein supplementation or diet can give, and we can some extent we can protect all these things. For example, type 1 diabetes is there. Every day they take the protein, that is the insulin, so they can survive. But in the case of epigenetics, environmental change, this environmental change, what it is doing is when you are eating high food, high fat diets, it is switching on the genes and switching off the genes. So when you stop, it will stop. Your physical exercise is gone down. Switching off, when you again you activate, switch on. Reversible, all these things. That's what no, obesity is not forever. Diabetes is not forever. When you change your style, lifestyle, definitely you can reverse it at the beginning stages. Of course, it is advanced or chronic, we cannot. But at beginning stages, we can. If you realize that one, yes, these are all changing. You must eat everything. That's why now people are cutting down the calories. What we have to cut down the calories? That also I'll come to that one, what I am doing here. So consider the twins. I said just now, the thing is except twins. When the twins are there, the best example I can give, I did not put my wife photo here. My wife is a twin. So both are the twins. My wife is a thyroid patient. Our clone is then, our sibling is no. So the thing is, she is a diabetic one. She got the diabetes in the age of 25. She got the thyroid problem in the age of 44. Why my wife is delayed all these things? The nutrition, the living condition. They live in the Dubai. So that is environment is different. Then my wife lives in India. So that no environmental effect is there. So that no, of course, identical twins are there, but their living lifestyle, their environment, their diet is having a role to decide your phenotype. That phenotype, you mean how you look. So the diet, exercise, exposure, all these things are deciding. One can become a different phenotype. So this is not, of course, identical twins also, both are genetics is same. Why one has become the diabetes mean? Yes, that is called epigenetical aspect. Your environment is changed, your lifestyle is changed, your living conditions are changed, your stress levels are changed. These are all important to know about your epigenetics. So to know about the example of this one, a study by Randy Jertil and all. So they have taken the genetically same animals, same rats they have taken. In that B12 deficiency, they are given in different doses. For this one is a control one, normal one. So B12 deficiency at some extent level, then they improved, increased. This is having a more B12 deficiency. It has become obese. What they did, they have taken the each rat and they cross it with the normal rat. After crossing with the normal rat, these are all females. When they cross it with the normal rat, whatever the pups burn, the pups also having the problem. They are also prone to the diseases. So that no, the deficiency, whatever is there, is it is having an effect on their offspring also. So that genetically same, but environment is changing. That is called, this is called epigenetical aspects. You can see the skin color also. The fur color is changed, you can see. It has become yellowish because this is a B12 deficient one. You can see the Randy Jekyll, you can put in the Google, you can get the paper. Here, what is happening in the epigenetic? We know the DNA language I said just now, adenine, guanine, cytosine, and thymine. In the case of the thymine, cytosine, the cytosine is undergoing for a one methyl group is adding here in this. Because adding of this tail, it is not allowing the enzymes to bind. So that no, for what one, this one methyl transferases are playing a role to add extra methyl group at this fifth position of the carbon ring. So in this we can see here, so that no, here it is adding in this one. Yes, it has become the methylated now. These are tails, we can see it is a normal one, we can see. To know more about this one and promoter region. So when it is a methyl groups are added, right? Not added. So there is no methyl group in this one. Promoter is normal. A gene is expressing and it's going well. Proteins are produced. So when a methyl group is adding here, so it is not allowing the enzyme to bind. So that it is not producing. Your gene is not expressing. There is no protein for it. So that environment is having the effect on the addition of the methyl group. In the case of the DNA, I'm telling. In the case of protein is different, different methodology is there. So that no process is different. But whereas in the case of DNA, only the methylation is taking place. So then methyl can be hypomethylated or hypermethylated. Methyl group can be added or methyl group can be deleted. Both way it is having the impact on the your protein or gene expression. So this is about the how epigenetics will work is only on the DNA level. It can be changed at promoter level. It is not allowing the enzymes to bind so that gene expression is stopped. So to know, come to my work again. So what I am doing in my lab. So just now, no, again at beginning, I want to refer your VC sir. Why is genetically different? We put the question here. Yes, genetic backgrounds are there. I want to give the same food for that. I want to give the same high protein diet or same high carbohydrate diet or mix of the both, keeping in view of the what population are eating. So that it is genetic background and environment both are working together or individually they are working. That's the question we put an interplay of this one. So then how the phenotype is changing. Then we studied all this. Thanks to DPT for giving me the funds. Then here we can see the rats I have taken, the genotypes are different. One to another one is different. Genetics of all the rats are different. I have taken 4 types of the rats. Their physical characters also varying from one to another one. So then I have taken all these things. I have given the what I said just now. For each group, I have given one group. I have given the high-fat diet for 100 days. I mean, that is called totally 3 weeks. I have 3 months. I have given 12 weeks. So one group I have given the high carbohydrate. One group I have given the mix of both. So how these are all effective? So then we can see the body weight gain. So body weight gain is changing from the high fat diet and high sugars. Mean HS mean high carbohydrate. Sugars I have given. HF is mean mix of both. Then we can see here only one rat strain is affecting more. That is a WNN. And this is a Vista rat is changing a lot. Significantly it changed. The total body weight also changes in the Vista rat. Not other rats are not affected. So which one is happening? In the high fat diet it is happening. In the case of this one, high fat and high carb, both are working here. So then body fat levels are changed. Your adiposity index levels mean among all the 4 animals, one animal is more prone to the diabetes because the genetics of that one is interacting more with that and all. So it is giving a lot of changes. So then we studied the tissue level, histopathology and all the things. This way we are working on this. Then this work. For that to know more, then we can conclude that work. It can be concluded that what you eat is what you are, and underlies the importance of genetic makeup of an individual vis-a-vis his and her responsible for environmental variable. So that now we did a lot of microarray, whatever the work. In that we identified several genes. So then some part of the work, how many genes are undergoing for this one, how many genes are changed, how many genes are underwent for the methylation and all. This is epigram, epic array we did. So some part of the work we published, still work is going on. So maybe soon we can publish that one. When come to the leptin gene promoter, another one. So another gene, is it that the leptin gene, one gene we have taken and we studied the methylation. So how the methyl is changing in this one. Then we can see here the red dots are there. At one position methyl group is adding, That's what it has become obese. One part of that one, not 100%. Yes, it is having the both genetic and epigenetic effect. Padma, just for a moment, my head is calling. I'll come in 1 minute.
Okay, sir. Dear participants, if you have any doubts or any clarifications, you can mention in the chat box so that we can clarify from the speaker.
So I'm sorry. Right, because now we are behind the schedule. That's what. Now we can go ahead. No problem. Now no more disturbances. Yeah, this is here the leptin gene we studied, and this work we published. Yes, because of this diet or genetical aspects. Mean genetics also playing a role for your methylation part. That what we understand in this study. So this, then come to this. Just now I said, whatever we discuss in this one is, it is coming from the mother womb, I said. So whereas, is it true? Only the culprit is the mother? Only the mother environment is important. Why not the father? So question has come for me. So it is a niche form is carrying this type of many memory. That's what now to rule out all these things. Then I put up the another proposal on that one. No? Maternal nutrition people studied maternal genes, fetal genes, paternal genes. So these are all studied. Whereas paternal nutrition, whatever the father is eating, father is a smoker, father is an alcoholic. So father is undergoing for this nowadays calorie restriction. They are also cutting down the calories because of living style and all. People are becoming obese. They are cutting down the calories suddenly. Bariatric surgery, etc. So many things are there. So that now another study we are doing is a paternal nutrition, cutting down the calories. Cutting down the calories is having any impact on the your child, offspring. That is work is going on. So one of my student is working on this one. So this way we can put the question. It's also environment. Your diet is playing a role. So that no, then this how it is impacting on the person who is cutting down the calories. One, and when they marry to a normal and healthy woman, so when they produce a children, their children are going to be affected or not? A pilot study showed that one. Yes, their children are undergoing for a change at early ages. So that is the idea to go for this one. Thanks to ICMR again for giving me the funds for this. Then here, in that when we are studying all these things, we identified, so of course that is already existing molecule FGF21, a molecule which is playing a role. This molecule having lot of functions on that one. So that we put the question in this. What exactly we have to do with this one? It is having many functions. How it is meticulously playing a role in the obesity to the pre-diabetic and pre-diabetic to the diabetic. Can we use this molecule as a medicine? Are the diagnostic marker in future for this metabolic diseases was our question. So that now we worked on this one. So FGF21, how it is playing a role. So then, so that FGF21 is playing the meticulous role in this one. A part of the work is published recently. Yes, it is playing a role in pre-diabetic to the diabetic condition. So then its role is very much important. So how it is impacting on the things, then pre-diabetic to the diabetes and obesity to the pre-diabetic we studied recently. This is about my work. Now at present I am doing two works. One is a paternal nutrition. The second work is ongoing on the FGF21. So with these few So slides and all, what I am trying to say is that what you eat is what you are. So friends, please eat seasonal fruits and vegetables. Then that is rich source of vitamins and minerals and your health. And also, so then whenever you find the time and whenever occasion comes, please plant a plant. Then thanks to all my funding agencies, ICMR, NAN, DBT, NH, and GCRF. I forgot to tell about my GCRF work. That is a stunting work. It's a big project. It is about 20 crores project. So that then I did not touch this one. Maybe next occasion I can tell. This is my team. So whereas of course after Padma left, this is one thing. So where people worked on this one. With these few words, then these things I can say thank you for all giving me this opportunity.
Thank you, sir, for your very effective and enlightening presentation. And you have started your lecture with a good message, that is our health, which is in our hands by consuming nature's gift, that is fruits and vegetables or seasonal fruits, etc. And also you gave the enlightened information and covered right from basics to the advanced technology and information. The genes and genomics, and you took us on tour of gene world and telling us that what are the latest software tools also can be used in this gene technology. You also discussed about the environmental factors, how they are influencing on the gene function, and now how metabolic disorders like diabetes and obesity can be controlled by our environmental factors. You also gave a good example of epigenetics on obese model. So thank you, sir. And your take-home message has been given as what you eat is what you are. Very, very effective and enlightening information and presentation, sir. And because I think there are no questions raised in the chat box, it is understood that everybody has been got the information. They are very understood. Thank you, sir. Thank you very much, sir, for your valuable lecture. Next, now I invite Dr. D. Sridhar from Department of Aquaculture to introduce our invited speaker, Dr. T. Anjana Devi, Madam, to the webinar participants. Over to Dr. Sridhar. Thank you, Rajendra, sir. Unmute, sir. Unmute.
Yeah, I leave for some time. Then I'll come back.
Okay, sir. Thank you. Okay, sir. Dr. Sridhar.
Ma'am,
Yeah, it is audible. Yeah. You can introduce
Okay,
our
madam.
invited speaker, Dr. Anjana Devi, Madam, to the participants.
Thank you, ma'am. Good morning to one and all. Honorable esteemed Vice Chancellor, Professor M. Jagannatha Rao, sir. Respected Registrar, Professor T. Ashok, sir. Respected Principal, Dr. K. Ramaneshwari, ma'am. Respected other principals, deans, officers. Respected invited speakers, Dr. K. Rajendra Rao, sir, and Dr. T. Anjana Devi, madam. Respected faculty members of the department, my dear colleague friends, and my dear students and other participants. Welcome you all for this national webinar. It's my immense pleasure to introduce today's invited speaker. Next invited speaker, Dr. T. Anjana Devi, madam, senior scientist from Applied Biology Department of CSIR IICT Institute, Hyderabad. She obtained her doctorate from Center for Cellular and Molecular Biology, CCMB, Hyderabad. Her research interests lie mainly in applying cell, molecular biology, and proteomics-based approaches to understand the underlying mechanisms behind autophagy, mediated mechanisms, and its role in cell survival and death of pancreatic cancer. She has guided many students, and presently 6 research scholars are pursuing PhD under her esteemed guidance. One PhD student is awarded doctorate recently. She has executed the projects as the principal investigator in the DBT sponsored projects and as a co-PI in the Indo-Russian jointly project and as a team member in the in-house sponsored ongoing projects, 11th and 12th 5-year plan projects. She got many research publication in peer-reviewed journals of international repute. She has presented her work in many international and national conferences, more than 25, edited books and contributed to 3 book chapters. She is a member of several prestigious scientific societies, Proteomic Society of India, Indian Science Congress Association, Indian Association of Cancer Research, Associate Fellow of the Telangana Academy of Sciences, member, Pancreatic Cancer Action, New York. With this, I would like to invite Dr. T. Anjana Devi Madam to deliver her presentation to today's national webinar participants. Thank you. Over to Dr. T. Anjana Devi Madam. Thank you, ma'am. Please deliver your lecture, ma'am.
Am I audible?
Yes, ma'am.
Yes, ma'am.
It's a prestige and honor for me to speak at this occasion and among the August gathering. At the outset, I would like to thank the organizers for giving me an opportunity to speak and present my research work at this virtually during this COVID pandemic. So, since the topic of the theme of the presentation is epigenetic modulators, so I thought that I would present one of the work recently. My student is awarded with PhD degree. So on the role of epigenetic modulator Cuisinostat in autophagy and differentiation in cancer, implications in therapy. Already BC sir and Dr. Rajender sir has given a very good introduction about epigenetics. And I don't think it will be better to more, there is no need for me to explain it again about the epigenetic changes or the epigenetic modulators function and all that. Anyway, briefly, I'll be speaking about the epigenetic modulator, and later I'll go on to the research topic, which recently we have completed in the lab. And also I would like to thank Rajender sir. He had been an external examiner for my doctorate student, and it was a privilege having him as the external examiner for the occasion. Yeah. So coming to the major area of research, the major area of my research is cancer biology, and the current research interests are cancer, cytoprotection, cell survival, cell death, epigenetic modulator, autophagy, and chemoresistance. As such, my lab has worked on 2 epigenetic modulators, mostly this SAHA and also Cuisinostat. And already I would like to thank Dr. Sridhar for giving such a good introduction. And I would also like to acknowledge the funding agencies without which this work would not have been possible. And it's very important to secure funding equally for carrying out the research work. So during from 2008 to 2000 till now, we had several ongoing projects in the lab where I was the principal investigator. One is the BioCare Award for Women Scientists. This is a special program for women where like it's biotechnology career advancement and reorientation program, which was the first grant awarded to me, and it was like a seed grant for me and helped me to establish the lab and also to carry out the work in the lab. And also another DBT funding grant came during that time, which was a young investigator grant. And both these projects have helped me to establish the lab successfully in IICT. Then I was also a part of the Indo-Russian joint project, and which was from 2017 to 19, and also a team member of the 11th and 12th 5-year plan projects. So from it was like 2011 to 2017, spanning this time. So all these funding has helped me to establish and carry out the experiments which we had planned in the lab. And during this time, I could also successfully guide project dissertation students, and one doctorate awarded, and presently 6 research scholars are pursuing PhD under my guidance. Yeah. So this is a general scheme showing the epigenetic regulation of DNA methylation, histone acetylation, and histone methylations, which are general epigenetic changes occurring inside the on the histones or the DNA, and these are regulated by different enzymes. And there are enzymes like writers, erasers, and readers which regulate these. And further, there are inhibitors which regulate these erasers, writers, and readers. So epigenetically, there are changes which are occurring, and at the same time, by using epigenetic modulators which affect DNA methylation or histone acetylation, all these things can be regulated. Am I audible?
Yes, ma'am.
Yes, madam.
You are audible, madam. Please.
Yeah, thank you. So coming to the neuroblastoma, it is a pediatric tumor of the sympathetic nervous system, and there are many. There are 3 major factors which are prominent in neuroblastoma, which is the tumor heterogeneity and minimal drug residue, and there is this drug resistance, multiple drug resistance. So to overcome all these things, we need to explore new therapeutic targets or new epigenetic modulators which can target and cure neuroblastoma. Minimal residual disease, even after treatment by surgery and other approaches, there are cancer cells, which some of the cancer cells which remain even after surgery, and they may relapse and develop again into the tumor. So these are some of the things which we need to take care. And the neuroblastoma itself is a very complex tumor. Giving a brief introduction about this, then these are the this is adapted from Childhood Cancer Association, showing different incidence of cancers among children, with maximum of leukemia 32%, CNS 26%, lymphoma 10%, soft tissue cancers accounting for 7%, and neuroblastoma accounting for 6%. Other tumors correspond to 5%, renal and bone tumor, and melanoma corresponding to 4%, and hepatic corresponding to 2%. On the other side, you see a table which shows the childhood cancer deaths per lakh. And here you can see that the percentage deaths of all these cancers. This is adapted from Journal of Clinical Oncology. So during embryogenesis, the neural crest cells, they can migrate, and they can differentiate into cells of different origin, cells of different forms. And then there will be a normal development of the neurons and the other cells. But in case this thing is not happening properly, there is a hyper neoplastic development occurring and resulting in neuroblastoma. So these things can be this can be targeted by using epigenetic regulators. And here we can use agents such as differentiation agents, and also there are reports that we can target using HDAC inhibitors. So based on all these studies, we have carried out work further. So there are 3 groups of neuroblastoma patients: the high risk group, the intermediate risk group, and the low risk group. The high risk group needs intense induction chemotherapy, and also sometimes combined with the reinfusion of hematopoietic cells and local radiotherapy. Excuse me. The intermediate risk group has can be treated by a milder chemotherapy continued by surgical resection. And the low risk group can be low risk group has a minimal chemotherapy, and some children are also cured by only surgery. So why we have chosen this molecule Quizinostat? It is also called as JNJ, and the complete details are given in the beginning, JNJ 2648181585. Why we have chosen this molecule is, it is a pyrimidine hydroxynate based novel second generation HDAC inhibitor, and it has a wide range of anti-cancer activity. It has been studied in other cancers, but no reports exist in case of neuroblastoma. So that was one of the key interests to choose this molecule for further studies. And it also works at a very low concentrations of nanomolar. It has excellent tissue distribution properties, and it is orally applicable. And it is also in clinical trials for several tumor entities. It is FDA approved drug, and it is also already in clinical trials. So these are some of the reasons why we have chosen this molecule. And no studies exist in case of neuroblastoma. So we have used this molecule for treating the neuroblastoma cells in vitro. In vitro is lab. The studies are carried out in the lab. So what was the hypothesis is that in the initial studies, when we treated the neuroblastoma cells, we have chosen two cell lines for neuroblastoma. One is the SKNSH and the other one is the IMR32. Both are of human origin. And these cell lines, when they were treated with this molecule, it has shown autophagy, and it has also resulted in differentiation. So we wanted to further go into details. As I earlier have explained, that the cells of the neural crest undergo differentiation, and if they undergo differentiation, there won't be any development of tumor, and the cells will be coaxed to normal cells. So that was one criteria of selection of this molecule. So since we got both autophagy and differentiation in the preliminary studies, we have chosen this molecule. We wanted to know the fate of the neuroblastoma cells upon treatment with Quizinostat. So Quizinostat or JNJ. So once we treated the cells with neuroblastoma, as I suggested, we got both autophagy and differentiation. And then it is also reported that there are these autophagy inhibitor molecules such as chloroquine, if they are used in combination with the existing chemotherapies, or also alone if they are used, they are also known to show some kind of therapeutic effects. So based on this, we have chosen chloroquine also for the study. And nowadays, during COVID scenario also, we have heard that the anti-malarial drugs, both chloroquine and hydroxychloroquine, were in vogue, and people are exploring it much for the treatment of COVID also. Though there were side effects and other things associated with it, people are trying to explore in detail. So, so based on this, both Quizinostat and chloroquine we have chosen. But again, we wanted to make it clear what was the effect using only Quizinostat on neuroblastoma cells, what was the effect using only chloroquine on the neuroblastoma cells, and what happens when I use a HDAC inhibitor Quizinostat along with the autophagy inhibitor chloroquine. Together, what is the effect on the neuroblastoma cells? So our hypothesis was what will be the fate of the neuroblastoma cells upon treatment with both these molecules. So as we all know, now we have another term coming up, autophagy. Am I audible and everything is fine?
Yes, ma'am.
Madam,
Yeah.
audible and fine, madam.
Yes.
Okay. So coming to the next term, autophagy. Autophagy is a self-eating process where the cells, it is a process of homeostasis where the cell organelles are recycled and they are later used by the cells. So in 2016, Yoshinori Ohsumi, a Japanese scientist, he won the Nobel Prize in Physiology and Medicine. And through his groundbreaking discoveries in yeast, where he has identified the autophagy and identification of the genes which were required for autophagy, the role of each of these genes was identified. And it was also identified that autophagy helps the cells to manage infection and malnutrition and any kind of stress which occur in the cells. So also it was shown that the lysosome degradation machinery, the role of lysosome degradation machinery and all this. So what is autophagy? Initially, it was only known as the vesicle nucleation, where the isolate double membrane is formed and then it engulfs the cellular organelles, recycle the cellular organelles, and it forms a autophagosome. This autophagosome then fuses with the lysosome. Lysosome is it contains the degradation machinery of the cell. So the lysosome and the autophagosome fuse to form the autolysosome, or also called as the autophagolysosome. And that's what is the entire process of autophagy. So during this, each of the steps, there are proteins which are involved, which play a key role, and I have listed some of them down. During the first phase, it is the ULK ATG1 complex, the PI3K complex, ATG proteins. There are several ATG autophagy related proteins, autophagy 1 to autophagy 14, which play a key role in this process. And then there is, in the second phase, the ATG12, the LC3 conjugation system, ATG5 to ATG12, and LC3-1 to LC3-2. These are microtubule light chain 3 protein, which plays a key role in this process. So, there is a dual role of autophagy which is occurring, and now there are many reports that autophagy can help in as a tumor suppressor or as a tumor promoter, and autophagy can act as a tumor suppressor in the initial stages where there is a less of stress and other factors. But as the tumor advances, it works in favor. The cells are, the autophagy machinery is hijacked by the cancer cells and it works in favor of the cancer cells. And therefore, autophagy can work in favor as a tumor suppressor where it can affect different factors in metastasis, chemoresistance, and cancer cells. Or it can act as a tumor promoter affecting metastasis, chemoresistance, and cancer stem cells. I'm not going into much detail. And then these are the experiments related to quisinostat or the J&J. So I have shown quisinostat as J&J in the slides, and we have checked the percentage. Yeah. So we have checked the effect of J&J on SKNSH and IMR32 cells dose dependently and also time dependently at 24 and 48 hours. And we observed that both J&J and chloroquine, when used individually at different concentrations, we have chosen concentrations of 5 to 1000 nanomolar in case of quisinostat, as it is the reported the reported concentrations are in nanomolar for J&J in case of other cancers. So that was the criteria for choosing these concentrations. And we also wanted to select the concentration which would work against the neuroblastoma cells. Again, we also have chosen chloroquine alone and checked its effect on SKNSH and IMR32 cells. And we found that both chloroquine and J&J had effect individually on the neuroblastoma cells, and they decreased the percentage cell viability in both the cases. And then then we have checked the effect of J&J and CQ together in case of SKNSH and IMR32 cells at 24 and 48 hours. And we found that when in combination, when both J&J and CQ are given, here CQ is given as a pre-treatment 1 hour and later followed by treatment of J&J. And we observed that upon treatment with both these molecules, there is a more decrease or more reduction in the cell viability when compared with individual individually treated cells. So both J&J and CQ, when given individually to neuroblastoma cells, there was little decrease in cell viability, but when they are used in combination, there is a more decrease in the cell viability. Then we also wanted to see the effect of J&J CQ co-treatment on morphology. What we observed is that when J&J or CQ alone are used, the cells do not get affected much, though there is a cell death. But upon co-treatment, there is a decrease, there is a change, and we observe that there is a vacuole formation in the cells. So we further wanted to check the effect. What are these vacuoles and why they have formed? So further investigations we have done. We also wanted to, not only the cell morphology, we also wanted to check the effect of these molecules on the long-term survival of the cancer cells. So both SKNSH and IMR32 cells, we have performed the clonogenic assay, and we observed that only when alone used, J&J and CQ did not show much reduction, though there was a reduction in cell viability in the colony survival rate compared to the control, but there was much reduction in the colony formation upon co-treatment with both these molecules. So we wanted to confirm what are these vesicle formation. So based on the theory, we understood that it might be autophagy, and we wanted to confirm that autophagic vacuoles are acidic vacuoles, and these acidic vesicle can be detected by use of acridine orange staining. So acridine orange staining is a green fluorophore dye, and at low pH it will fluoresce and give a orange fluorescence. So when we treated the cells SKNSH and IMR32 with increasing concentrations of J&J, we observed that there is an increase in the reddish orange fluorescence in the cells. You can observe at 50, and it is increasing at 100 nanomolar and 200 nanomolar in the green cells. And also in the IMR32 cells, SKNSH and IMR32 show increased acidic vesicle formation upon treatment with the increasing concentration. So in order to further reconfirm that it is acidic vesicles only and it is the autophagy only, we have also done this MDC staining, and MDC gives a increased fluorescence around the cells, and you can see that compared to control at 50, 100 and 200 nanomolar treatment of J&J, you observe an increase in the fluorescence in the form of dot particles with increased fluorescence around the cytoplasm. So both you can observe in SKNSH as well as IMR32 cells. So we confirmed that it was autophagy. Also during the autophagic process, LC3, the microtubule light chain 3 protein converted from LC31 to LC32, and LC31, the cytosolic form 18 kDa, is converted to the LC32 form, which is 16 kDa, and by the addition of phosphatidylethanol, it becomes a membrane bound form. And this LC3 conversion also we have checked at different concentrations of J&J, 50, 100 and 200. We observe that there is an increase in the LC32 levels, and we also have taken rapamycin as a control. Rapamycin is a known autophagy inducer, and we have chosen this molecule, and we observed that there is an induction of autophagy upon J&J treatment. We also wanted to check the effect of J&J on autophagy flux. So when during the autophagosome formation, there is a LC32 increase, and autophagosome then fuses with the lysosome to give the autolysosome, and then autolysosome degrades to release the cellular organelles, which can be recycled for use. So during this autophagosome to autolysosome conversion, if we use autophagy inhibitor, it will block the lysosome fusion with the autophagosome, and there is also a LC32 accumulation. So to check that, we have used the autophagy inhibitor bacillomycin A in our experiments, and we observed that there is an increase in the levels of LC32, which shows that there is a upon treatment with the autophagy inhibitor, there is an increase in the autophagy flux upon treatment with J&J. So again, we wanted to check the effect of J&J and CQ on caspase-3 and PARP levels. Caspase-3 and PARP are known apoptosis markers. So I have been speaking about autophagy, and then I have been speaking about the effect on both cell morphology, clonogenic assay, and all this. So the main take-home message is that upon treatment with J&J, we got autophagy, and when we treat with CQ, there is the autophagy is stopped and we get apoptosis. So the detailed mechanisms are I will be showing at a later stage. So upon treatment with CQ, we have checked the levels of cleaved caspase-3 and cleaved PARP, and we observe that there is an increase in the levels of cleaved PARP and cleaved caspase-3 upon treatment with J&J and CQ. As you can see from the densitometric graphs more clearly. So caspase-3 and PARP are markers of apoptosis, and the levels increased upon treatment, upon co-treatment with CQ. However, when we use a JEDVAD FMK, the caspase-3 inhibitor, we observed a decrease in the levels upon treatment with in the co-treatment process. So we also checked the effect of J&J CQ on ROS production, and we observed that J&J CQ co-treatment results in increased ROS levels, and we also have taken hydrogen peroxide as the control, treated cells as the control, and in presence of the ROS scavenger, there was a decrease in the effect. We have also checked the effect of J&J CQ co-treatment on the mitochondrial membrane potential, and we observed that there is a disruption of the mitochondrial membrane potential in the both SKNSH and IMR32 cells. So our study shows that J&J or Cuisinostat induces autophagy in neuroblastoma cells, SKNSH and IMR32. The Cuisinostat mediated autophagy inhibition by chloroquine pre-treatment sensitized the cell viability of neuroblastoma cells, and autophagy inhibition by chloroquine increased LC32 accumulation, increased ROS, and consequently decreased the mitochondrial membrane potential. So this suggests that J&J CQ co-treatment represents a promising new therapeutic approach for neuroblastoma treatment. Well, so all these studies we have recently published last year during the lockdown in molecular and cellular biochemistry, and this is the published paper. Then we also wanted to understand the effect of Cuisinostat on differentiation. As I earlier suggested, that we also observed some clues which showed that it may be a differentiation, and if there are differentiation defects, there is formation of neuroblastoma. But however, if we treat with differentiation therapy agents such as retinoic acid or any other differentiating agent, it can lead to the differentiated neurons. So these are some of the listed differentiation agents: retinoic acid, vitamin D, PPAR ligands, hormone receptor ligands, and SAHA, vorinostat, etc. So the well-known agent used for differentiation is retinoic acid, but still more than 50% of the treated patients still develop recurrence, and therefore there is an essential need for development of identification and development of novel alternative differentiation agents to treat the resistant patients. And discovery of new differentiation agents has been very slow. So these were some of the interesting points which we wanted to study. So we have chosen again Cuisinostat, and I wanted to understand its effect as a differentiating agent in neuroblastoma. So what we have done is we treated the cells and we observed the morphology of the cells upon treatment with JQ at different with J&J at different concentrations. So we observed that there is formation of neurites in the microscopic observations, and we also measured the average neurite length, and it was clear that there is a differentiation occurring. And in order to also make it sure that it was the differentiation process which is occurring, we have also measured the levels of the differentiation markers beta tubulin 3 and enolase 2, and we observed that upon treatment with J&J, there is an increase in the levels of these two proteins, which clearly indicated that the cells are undergoing differentiation. We have also chosen retinoic acid as a positive control, but somehow it did not show any good effect, but still we can observe some change in the levels of beta 3 tubulin and ENO2 at 48 hours when compared to the control. We also have performed the clonogenic assay at different time intervals in SKNSS cells, and we found that dose and time dependent inhibition of cell viability. And we also, since it is the HDAC inhibitor, we have also checked the levels of H4 acetylation levels, and with the histone levels as the control, H4 and H3 acetylated levels. Also, we have observed the levels of HDAC, histone deacetylases 1 and 2 in the studies, and we observed that there is a decrease in the levels of HDAC upon J&J treatment. We have also observed the effect on cell cycle, and we observed that there is a G1 arrest which is occurring due to the effect of J&J. And cyclin D1 and CDK4 are important proteins of the cell cycle phase, and the arrest was confirmed by checking the levels of these proteins. So when we treated, we have also performed one of the experiment where we have used the autophagy inhibitor CQ, and we observed that when we treat with autophagy inhibitor CQ, though LC32 levels increase, there is a decrease in the levels of beta tubulin 3, which is a differentiation marker. And based on this, we confirm that autophagy occurs first before differentiation, and autophagy is required for the differentiation to proceed. This work was accepted 2 days back in molecular biology reports, and we also need to further explore in detail about the studies in vitro as well as in vivo. So these are the details of the kind of experiments we do at IICT. And this is my small group. Actually, one of the student is missing who has taken the photo. And so this is a small group at IICT, but now I have 3 more students, so total 6 students. And this guy is the first PhD student who has recently completed. And I would like to thank Director CSIR IICT for providing all the infrastructural facilities and all the facilities at the institute. And I thank all the funding agencies, the DBT, DST, and UGC, and also the fellowship funding agencies to my students. And I also thank my team, without which this work is not possible. And I thank all my collaborators who have been very supportive and who have been very supportive for carrying out the research work. Thank you.
Thank you, madam. Thank you, Dr. Anjana Devi, madam, for your effective presentation. And we learned from your lecture as that the incidence of the childhood cancer incidence, and you emphasized on the therapies for the neuroblastoma disease, how the quinostat is acting as the therapy, as well as the both in combination of J&J and chloroquine, that is the co-treatment, how they are affecting the anti-cancer or neuroblastoma differentiation. And also you have enlightened us about these factors are acting as a epigenetic modulators in treating the cancer, which can be acting as one of the cancer therapy. So it is very, very effective and informative lecture, madam. Thank you, madam, for accepting our request and invitation and sparing your valuable time, ma'am.
Thank you.
Thank you, ma'am. Now I invite today's co-patron, Principal, University College of Science and Technology, Arikavinana University, Dr. Ramneshwari Madam, to propose the concluding remarks to the webinar. Madam, Ramneshwari Madam. Vijayanirmala Madam.
Padmavati Madam,
Yes, ma'am.
I think you can go to the next program. Madam, I think she has left, I think. Maybe some
Madam,
net
just
problem.
to be brief,
You can go to
one or two.
thanks.
Okay.
Myself.
Huh.
The senior most faculty of the department, Dr. Matareddy sir, I request you to give small message and give the concluding remarks. Matareddy sir. Okay. So thank you very much. Okay. Matareddy sir.
Hello.
Sir, good afternoon, sir. Please.
Can you hear me?
Yes, sir. Yes, sir. Please go ahead.
Yeah, we
Sir?
had
Yes, sir. Thank you, sir.
lectures.
Yeah.
Today's lectures are wonderful. Dr. Rajendra Rao and Dr. Anjana gave very good lectures, which are useful to our students very much. And I thank both of the experts who gave expert lectures today. Thank you.
Thank you, sir.
Madam,
Thank you very much, sir.
Question from the chat box, ma'am. One question from the chat box. And also, the question is that what was the differentiation percentage after the treatment of retinoic acid? It was asked by Dr. Bujjibabu Chiripi. What was the differentiation percentage after the treatment of retinoic acid?
The question is to Anjana Devi, madam. Okay.
Anjana Devi, madam.
Yeah. We have observed differentiation, and the percentage exactly I'm not sure, but there was differentiation. As we observed from the studies, we observed that there is at least 60% differentiation among the cells. Yeah, that was the thing, and we further also confirmed with beta-3 tubulin and the enolase markers of differentiation.
Okay, ma'am.
And there is a variability.
Sir. Madam, Ramneshwari madam di unmute అవ్వట్లేదు. మేడం ఉన్నారు.
నేను చేయనా? చేస్తాను. నేను చేస్తాను.
చేయండి మేడం ఒకసారి రామేశ్వరి మేడం ది.
చూస్తున్నా.
Now it's working, madam.
Okay, madam. Madam Ramneshwari madam, please we invite you to present the concluding remarks, ma'am.
Hello, can you hear me?
Yes, ma'am. Yes, ma'am. It is audible.
Yes. A very good afternoon to one and all. Concluding remarks, it is my privilege to do it on behalf of the department, being a member of the Department of Zoology and also as a principal, University College of Science and Technology. And first of all, I would like to congratulate the head and the convener of this webinar and the co-convener, Dr. IGN Padmavati, who has been instrumental in bringing in the resource persons, and that too from two different national institutes, National Institute of Nutrition and National IICT, Indian Institute of Chemical Technology. Thank you, Dr. Rajendra Rao sir and Dr. Anjana Devi madam for agreeing and coming over here readily to share your research work to our students. And I congratulate the department and the head of the department who has taken a lead in organizing the webinar. And as Rajendra sir has mentioned, sir, you have taken us back to the Mendel's laws, the very basic fundamentals of it. And I hope the students and the participants have benefited. Just in a simple sentence to put it, the one gene, how it works in different individual organisms of the same species. I think you have put it in a very simple way to understand, and this would help our students currently who are pursuing a program in different papers also that they are studying, like we have the genetics and evolution part of it, and also we have principles of ecology, the population genetics, so on like that. Thank you very much, sir, for delivering your lecture here. And you also have shared your work, like what you are doing, because I know many people do not share the work that they do completely, but you have done with your personal, and you have quoted your personal experience also throughout the lecture. Thank you, sir. And thank you, Dr. Anjana Devi madam. Though I'm not into that your area of specialization, but what I could understand is that you are doing the cutting edge research in the what you say, cancer therapy. And I hope that some of our students could take up some type of project work, or they can go ahead in their research to in this particular area, which is of immense usefulness to the human beings and the society at large. And now I would like to convey my thanks to our administrators, especially our Honorable Vice Chancellor, Professor Jagannath Rao Garu, who takes a keen interest in all the academic activities of all the what you say, colleges and the departments, because he says whatever we do it should be beneficial to the students. The students should be getting some type of benefit out of it, and whatever. And I think you heard about his message. He does not just come and address, but he does a thorough study on the subject, and he puts forward his ideas also. And from his ideas also, we get certain different dimensions to look at that particular topic. And I thank our registrar for providing all the infrastructural support together with the team of the webmaster for providing all these facilities to make this webinar a grand success. Thank you one and all for attending this, the participants, and hope the students take back something so that they can look forward what they can do further ahead in their careers. Thank you one and all. Over to Padmavati madam.
Thank you, madam. So for briefing the webinar presentation, and also thank you, ma'am, because you are the one behind who is pushing us and encouraging us to arrange such type of course. Main person is our professor, Honorable Vice Chancellor Jagannath Rao sir. At the same time, you are also insisting us to conduct and arrange this type of webinar so that which will be helpful for our students, and they may get encouraged to enter into the research field, just not to stop at their PG level. So I also thank our invited speakers also, who has spared their valuable time and presenting a very effective and informative lecture. Now, I invite Dr. Murli Mohan sir to propose vote of thanks.
Yes, ma'am. Thank you, madam. Am I audible?
Yes, sir.
Thank you, ma'am. Good afternoon, one and all. It's my privilege to propose vote of thanks. My immense pleasure to convey my sincere gratitude to our Honorable Vice Chancellor, Professor M. Jagannath Rao sir, for encouraging us to conduct this type of webinars in order to explore various research areas and generate awareness among the participants, especially our students, to get motivated to enter into research field after their master's program. I express my sincere thanks to respected Registrar sir for giving this opportunity and all the logistic support for conducting this webinar program. I would also like to thank our invited speakers, Dr. K. Rajendra sir and Dr. T. Anjana Devi madam, for their acceptance to deliver the presentations on epigenetic approach, sparing their valuable and precious time despite of their busy schedule. And thank you, Rajendra sir and Anjana Devi madam, for enlightening all of us with your effective and informative presentations. I also convey my sincere thanks to our today's co-patron principal madam, University College of Science and Technology, Dr. K. Ramanashiri madam garu, briefing the webinar and presenting concluding remarks. I convey my sincere thanks to our today's convener and head of the Department of Life Sciences, Adigavi Nannaya University, Dr. P. Vijay Nirmala madam garu, who is instrumental in conducting this program. And also, thank you madam for giving me this opportunity as well. I also thank all the faculty members and colleagues for sparing their valuable time and attending this program. I also thank all my dear students and other participants, supporting staff, webmaster especially, for supporting this platform, this online platform, press and media of Adigavi Nannaya University for attending and supporting the program, making it a grand success. Thank you all, and once again, thank you for this opportunity. Over to you, madam. Padmavati madam.
Thank you, sir. Madam, over to Dr. P. Vijayanarma, madam.
Thank you, Dr. Padmavati madam, for your for hosting this in a very smooth manner. I thank once again. I thank the higher authorities and my faculty members and non-teaching staff for their support in organizing this. A special thanks to the two speakers, Dr. Rajendra Rao sir and Anjana Devi madam, for sparing your time and being with us today and sharing your knowledge with us. Thank you, one and all. And my our senior faculty member, Dr. Mataridi sir, thank you, sir. And we have our student here from USA, Dr. Bujji Babu, who had attended this. Thank you, Bujji Babu, for joining this. Being you have proved that you are the students from here and you are extending your services whenever we are asking. Thank you. I thank once again Dr. Aijan Padmavati and Principal Madam Dr. K. Ramaneshwari and Honorable Vice Chancellor and Registrar sir and one and all. Thank you, one and all. The webinar is ended and you can leave the meet. Thank you.
Madam, participants are asking for the feedback form.
Webmaster, Srinivas sir. Srinivas garanడి.
Sir, madam.
కొంచెం షేర్ చేయరా అది ఫీడ్బ్యాక్ లింక్?
Sure, madam.
థ్యాంక్ యూ అండి. అది చాట్ బాక్స్ లో పెట్టేస్తే వాళ్ళు ఫిల్ చేసి సబ్మిట్ చేస్తారు.